Welcome to Lesson 1 of Module 6, Genetic Change. In this video, we're going to be looking at mutations. And the syllabus points we're covering today is the inquiry question, how does mutation introduce new alleles into a population? We're going to look at mutagens, the different types of mutations, and then the significance of them and how they affect organisms. When we talk about a mutation today, we're talking about any changes that occur in DNA. A mutagen is a factor that induces mutation, and they can be either chemical, physical, or biological. There are somatic mutations, and they occur in body cells and only affect the individual that they occur in. And then there's germ-line mutations that occur in germ cells, which are our sex cells. They're heritable and therefore can be passed on. When we're trying to work out what type of mutation is happening in our DNA, we have these five questions we need to ask. Once we have our answers to these, we can determine the type of mutation which we're observing. So we need to know the cause of the mutation, how much has changed in the genetic material, the effect on DNA, the effect on the phenotype, and whether these mutations are heritable. Mutations can fit into one of these three categories, so they can be neutral and have no effect on survival. They can be harmful and decrease the likelihood of survival, or they can be beneficial and increase the likelihood of survival. The first example which we're going to look at are point mutations, and these are mutations that alter, add, or remove only one or very few nucleotides, and it's usually affecting a single gene due to how few nucleotides it plays with. So these point mutations can either be substitutions or they can be frame shifts. So substitution is where one nucleotide is replaced by another, and they can either be silent substitutions, missense substitutions, or nonsense substitutions. So silent results in a new codon, but this codon codes for the same amino acid. There's a missense, so it results in a new codon and a different amino acid, and a nonsense results in the creation of a stop codon. So we can see how these all would affect the functionality of the gene. And then there's a frame shift mutation, which involves one or two nucleotides being added or removed from the sequence, and therefore every codon in that sequence from that point forward is changed. Then we have block mutations, and these affect large sections of chromosomes and therefore usually multiple genes. And these usually occur during meiosis. So we can have duplication, where we have multiple copies of the same gene, inversion, where we see a piece of the gene breaking off, rotating and reattaching to the same chromosome, deletion, where we remove a section of the chromosome, insertion, where we see the chromosome breaking off and attaching to a different part of the chromosome, and then translocation, where there's a whole chromosome or a section of chromosome becoming attached or exchanged with another chromosome. So here we have some arrows here which are showing us all the different types that we've just been through. So we can see how when we have the section of that chromosome, how depending on the way they move and where they end up going is the type of mutation that we're seeing there. We then have mutagens. So chemical mutagens can be a few different chemicals that affect the sequence of DNA. So we can have the intercalating agents, where they alter the shape of the DNA by inserting themselves in between the bonds. We can have base analogues, which chemicals are structurally similar to the bases in DNA, so they're incorporated, but therefore, because they're not nucleotides, the DNA no longer functions. And then we have DNA reactive chemicals, which can react directly with the DNA and cause breakages or cross-links. So the higher the exposure of these chemical mutagens, the greater the risk of the mutagenic effect. And a chemical mutagen can also be carcinogenic chemicals, which are things like tobacco, arsenic, and mustard gas, which can also affect the structure of our DNA. Then there are physical mutagens. So we have particle radiation, electromagnetic radiation, ionizing radiation, non-ionizing radiation, and therefore we can then use these sometimes in medicine where we purposely damage DNA. So all of these have the energy being emitted and therefore changing the structure of our DNA. And this is how we use radiation to treat cancer cells, causing their death. And then we have biological mutagens. So these are living molecules that can interfere with the functions of oncogenes or tumor suppressor genes. Viruses, such as retroviruses, they can insert themselves or their genetic information into the chromosome of the host cell, and therefore cause cancer. And then we have transposable elements, which are short sequences of DNA that move around in the genome. Because they constantly move, they're called jumping genes, and therefore they can cause errors in replication and interfere with their functioning. Then there are intron mutations. So introns from Module 5 are non-coding regions. So therefore, mutations in these sections can usually have no effect on the organism. However, if they occur at the splicing site, they can create a pseudoexon and therefore read the intron as a coding sequence. And then if there are any intron mutations that create new splicing sites, we can see a reduction in gene expression. And this is what happens, in fact, in cystic fibrosis. And then if there are mutations in any promoter or terminator regions, we can see an over or under expression of a gene, and therefore the amount of gene product being produced can be either increased or lessened, and therefore have effects on the organism. We can then have non-coding RNA mutations, and therefore, if they occur in any of these, it affects the functioning of a cell. And depending on where it results in the RNA, it can usually cause death. And our last two slides, we're just going to compare somatic and gametic mutations. So somatic mutations occur in somatic cells, and we know these as body cells, and these usually occur during mitosis, the way in which these cells grow and divide and replicate. Since they are postzygotic, they occur after fertilization. We all experience a low level of this as we grow in age. The more errors we have in our DNA, the more that we can see abnormal growth of tissues as we get older. And then mosaicism is where there's a mutated genome that can be carried in a few cells, and it's where we see sometimes people having different pigments present in their skins due to this mutated genome. Then we have our germ-line mutations, and these occur in reproductive tissue and occur in our gametes. So therefore, we can have defective gametes resulting missing, extra, or malformed chromosomes. Our body has a good way of repairing this DNA in these tissues. However, they still do happen. One of the examples is non-disjunction, and this is where we see a chromosome not separating from the other during meiosis, and this is what we see happening when someone inherits Down syndrome. So we can see here, instead of our gametes all having one chromosome, some of these have not disjoined properly in the right place, so we can see a change in the gametes producing chromosomes. That includes lesson 1 of module 6. Make sure you tune in for lesson 2.